Hello, students. So, welcome to Learnosphere. This is Anne Maria de Cruz, and today in this video, we are going to study the chapter called the fundamental unit of life. So, mostly we'll study about the cell. Okay, what is a cell? Okay, its function, its components. So cell will have its own organelles in it, right? So each function of its organelles we're going to study and its importance. Okay, so let's start with the lecture. Okay, now before studying what is a cell, we need to know how exactly the cell was discovered. Okay, so the cell was first discovered by Robert Hooke. Okay, he's a scientist, and cell was first discovered by Robert Hooke. Okay, in the year 1665. Okay, now how did he discover? Basically, he took a small piece of cork. Okay, he took a small piece of cork, okay, or a slice of cork. Now, what is cork? Okay, so in this diagram, you can see. Okay, this diagram, the outermost layer of the bark. Okay, you have seen a bark. Okay, you have a tree. Okay, so tree basically, if this is a tree, okay, now this is the trunk of the tree. Okay, the trunk will have the bark. Okay, that outermost layer of the bark he took. Okay, basically he took this part which you can see. Okay, he took that part, he took a slice of cork. Okay, from the trunk of the tree, from the bark of the tree, he took a slice. Okay, slice of cork, and then he put it under the simple magnifying device. So in those days, in we're talking about 1665. Okay, there we did not had proper microscopes. Okay, microscopes were very proper, nowadays, which the microscopes which we get electronic microscope, a lot of various microscopes, but there then in 1665 there were no much very well developed microscopes. So basically they were simple magnifying devices. So Robert Hooke, what did he do? He took a slice of cork. Okay, cork. I hope you understood that it is the bark. You have seen the bark, the outermost in the trunk, the outermost layer is there. That is bark. Okay, inside the bark, the inner layer of the bark. Okay, that is called cork. Okay, in the diagram you can see properly which is cork. Okay, basically he took the slice of that cork and he then observed under the simple magnifying device to know how it is exactly made. Okay, now after observing through a microscope, what did he observe? Okay, so he could see this. Okay, he could see under a simple magnifying device, he could see something like this. Okay, now in by looking into a diagram or by looking into this, what he concluded is it looked like a honeycomb. Okay, he said like it looked like a honeycomb, or it looked like small boxes. So in the diagram you can see there are small boxes, right? So for him it looked like this. Okay, like small boxes, or he said it looked like a honeycomb. Okay, and then he said that each box was partitioned but was separated by a wall like thing. Okay, you can see some boxes here. Okay, so these boxes were separated by a wall. Okay, each box is separated, or it is partitioned, or it is partitioned by a wall. Okay, so then he said that I will call it as a cell because it is looking like a box. Okay, in jail and all, it's if you're seen in movies and all, there's a room. Okay, empty room. There's nothing. So we call it as a cell. Right. So exactly, Robert Hooke also saw kind of a very empty boxes, okay, which is partitioned by a wall-like thing, and he said, okay, I'll call it as a cell because it's looking like a cell to me. Okay. Now, um, okay. So yeah. So what did he notice? He noticed partition boxes. Okay, when he observed the slice of cork under a magnifying device, he got to see some partitioned boxes or some compartments. Okay, compartments or partitioned boxes. He said that these boxes appeared as a honeycomb. So in the diagram also you could see it was looking like a honeycomb, right? So these boxes, these group of boxes were looking like a honeycomb. Okay, he also noticed that one box was separated from the other by a wall or partition. So these boxes were separated by a wall or partition, and he said he called these partitions or these boxes as cell. Okay, so basically for him, when he observed the slice of cork under a magnifying device, he could see some boxes, okay, which is partitioned by a wall, okay, and it seemed it looked like a honeycomb to him, and he said, okay, I will call it as a cell. So that's how he introduced cell. Okay, but in these boxes, if you see, I'll show you the diagram. Yes. So in these diagrams, if you see, these boxes are empty. Okay. If we are looking into a live cell, okay, a live cell, along with the cell, along with the wall, now we call this wall as cell membrane. Okay, this wall which is talking about, it is partitioned. Each box is partitioned. Which wall is partitioning each cell? It is cell membrane. Okay, now basically, in if these cells were alive, the organelles also he could have seen. Okay, these organelles inside, there will be nucleus. You will study what are the organelles. But in these boxes, in these cells which he observed, it was empty. Okay, it was empty. So he said, okay, these cells are dead. Okay, so the slice, when he observed the cells of the cork are actually dead. So that's why you can only see the wall. You cannot see anything inside. Okay, so he also said this that these cells are dead because I'm not able to see any organelles inside it. It is completely empty. Okay, so he said these are dead cells. But if you see in living cells, you can also see nucleus, you can also see the organelles, okay, associated with the cell, right? Okay, so now how can you see, okay, if you're in a lab, okay, and you have to see cells, okay, from the living organism or a living thing, you want to see cells. Okay, so how can you see cell? Okay, so basically you take an onion. Okay, everyone has seen an onion, yes, and you have to peel off the onion, the outermost dry part which is there, you're going to peel it out. Okay, that edible part is there. No, that edible part you're going to take, and you're going to try to take the outermost layer of the onion. Okay, outermost transparent layer of the edible part is there. No, that edible part, the transparent layer, you have to try to take. Okay, you will take a onion, peel it off, the edible part will be left. There's no need to cut it or something. You have to try to take skillfully the outermost transparent layer. So when you try to peel it off, if you get that transparent layer, you're successful. Okay, so try to take the transparent layer of the onion. Okay, then what you're going to do, Just give me a second, I'll take my pen. Okay, so basically you will take that onion peel. It should be transparent. Okay, then you're going to put saffron stain. Okay, you're going to put a stain called saffron. Okay, saffron. Okay, and why you're putting a stain? Okay, if you have to see the cells properly, okay, clearly, effectively, so that's why we put the stain. Okay, it is pink in color. Okay, it is light pink in color. So if you put the stain, you'll be able to see what is inside the cell properly. Okay, so you will put a stain called saffronin. Okay, to this onion peel which you have taken in the petri dish. Okay, you're basically it's a petri dish. Okay, it's called petri dish. Okay, now what you're going to do, you have put the stain, you have put the stain. Okay, now keep the stain as such for some time. Okay, for 1 minute you put the stain. The peel should properly absorb the stain. Okay, then afterwards what you're going to do is you're going to rinse the peel which you have stained. Okay, you have to take out that excess stain, right? So a little lightly, you're not going to wash it very roughly, you're going to rinse it. Okay, lightly rinse the onion peel. Okay, rinse it. Okay, so the peel will have very light pink color. Okay, it is a transparent peel. When you try to take, it's a transparent peel. You put a saffron in stain. Then you're going to rinse it with water. You only very little stain should be left out. Okay, you have to wash it in such a way only very little stain should be left out. Now, what you're going to do? You're going to take this peel and you're going to put it under the slide. Okay, you put it under the slide. Okay, you put it under the slide. Then what you're going to do is you will on top of that peel which you have put under the slide. Okay, you are going to put glycerin. Okay, don't pour the glycerin, put few drops of glycerin on top of it. Okay, glycerin you're going to put. Okay, now this is called this is called cover slip. Okay, the small square thing which is there, no, that is called cover slip. Okay, now what you're going to do after you put 2 drops or 1 drop of glycerin, you're going to cover it with a cover slip. Okay, you're going to slowly put it using a cover slip. Okay, now when you're covering this peel with a cover slip, you have to make sure that there should be no air bubble formation. There should be no air bubble formation, otherwise it will, when you're trying to look under the microscope, you will be not able to see it properly. The organelles whichever is there, you will be not able to see it properly. Okay, so for that you have to put it in such a way that there should be no air bubbles. Okay, then finally put it under the microscope, arrange the light of the microscope. Okay, under there's a mirror. If you have seen in the lab, arrange it under the microscope, and then you'll be able to see the cells of the onion. Clear? Okay, so first what you're going to do, if you want to see the cells of the onion, first what you're going to do is you're going to peel the transparent layer of the onion. Okay, then you will collect it in the petri dish. Later, what you're going to do is you're going to put some stain. Okay, which stain you're going to put? It is called saffron stain. Saffron and stain. Okay, you're going to put stain. You're going to rinse the excess stain. Okay, you have to rinse in such a way that only very little stain should be present in the peel. Okay, then you're going to put it under the slide. Okay, then afterwards you're going to put few drops of glycerin. Okay, then you're going to cover it with a cover slip. Okay, when you're covering it with the cover slip, there should be no air bubbles. Then you're going to and then you have to excess glycerin which has come out from the while you're covering it with the cover slip. After covering it, there can be excess of glycerin on the slides. Okay, there can be excess of glycerin on the sides of the cover slip. So by using the blotting paper will absorb all that excess glycerin, and it will make your slide very clean. Okay, that excess glycerin, if it's there, it destroys the slide. Okay, so in order to make it clean, the excess of glycerin which has come out, by using a blotting paper you will clean it off around the sides of the cover slip. Then you're going to observe it under the microscope. Okay, right.